Let's begin with the next chapter of general microbiology, and that is bacterial genetics. This has not only topics pertinent to bacteria, it will also help you towards the end in some important topics with regard to humans, and that is CRISPR-Cas9. Let's get going. Let's start with our very basic. If I have to ask you the difference between a DNA and an RNA, the first thing that you'll say is deoxyribonucleic acid. So it's going to have a deoxyribose sugar entity, whereas when I talk about RNA, that is going to have only a ribose sugar entity. What's the next difference? The difference is between the nucleotides. Both of them have two purines and two pyrimidines. I think this all of you know that the two purines are going to be A and G, and the pyrimidines are going to be C and T. And how are they held together? They are held together by the hydrogen bonds. However, when you go to the RNA, you again have the same A and G, but what happens to C and T? It now becomes C and U. So can I say that uracil is going to come and replace thymine? That is important. So remember once again, when you're talking about the DNA, we have AG and CT, purines and pyrimidines. When we are talking about the RNA, we have AG and CU. That's a very basic thing to start off with. Let us start off with the first part, and that is mutations. Firstly, you'll have to tell me what is mutation. So mutation is a permanent change. Permanent, that is very important. If you want me to add few words to it, it's gonna be permanent irreversible change that occurs in the DNA. It's a permanent irreversible change that occurs in the DNA. So how many types of mutations do we have? And once it happens, it's never getting back. So the first type is referred to as a point mutation, and the second one is known as a frame shift mutation. So I'll explain them one by one, and this will help you in your pathology. I've taught you in genetics of path and humans also. So talking about point mutations means that the problem is going to happen at one point. So that could be transition. So probably at one point, purine is replaced by a purine. So it's between A and G. Or pyrimidine is replaced by pyrimidine. So basically at one point, replacement is by the same family member. So that is going to be transition. However, if it's a cross family member thing, means a purine is replaced by a pyrimidine, that is going to be transversion. So I'll give you a very basic and a very quick hint. For example, if this is a kind of a DNA sequence, random DNA sequence that I've written, and I say over here, instead of G, now over here, if A has come, so you will say, okay, instead of one AG, same family, na purines, instead of one, another purine has come. Purine has been replaced by purine. You will say it is transition. But over here, if I say a C has come, then you will say this means purine has been replaced by pyrimidine. That becomes transversion. So whatever the case is, it's at a single point. Please note that a point mutation can actually be having three types of point mutations we have. The first one is known as a silence mutation, means it happened but it did not result in any change. It has not resulted in any effect. On the other hand, if there is a change that happens, either that could be a missense mutation or it could land up in a nonsense mutation. And I'm also teaching you this not only with regard to bacteria but also with regard to us. So I'll tell you, see, you say that ma'am A got replaced by G, but it made no difference on the bacteria or the human beings. I'll say very good, it's a silence mutation, no one got to know about it. But if I say missense mutation, that basically means that, let's take an example of, everyone's heard of sickle cell anemia, yes? What happens in sickle cell anemia? At beta 6 position, glutamate is going to go away and valine is going to be welcomed. Glutamate is going to go away and valine is going to be welcomed. One amino acid goes, one is going to come. As a result, what happens? Adult hemoglobin changes to what, guys? Adult hemoglobin changes to sickled hemoglobin. So has the sense changed? Of course, this is a missense mutation. The sense has changed, which means the adult hemoglobin has changed into sickled hemoglobin. The entire story has changed. The sense has changed. Versus if I'm talking about a nonsense mutation, I'm sure everyone has heard of beta thalassemia, very famous anemia that we have. In beta thalassemia, is there any beta chain production? No, everything has stopped. Nonsense. Hemoglobin production has stopped. See, look at the difference. Over here, there is no beta chains, means there is no hemoglobin production happening. So over here, everything has stopped. In sickle cell anemia, hemoglobin production was happening, but it's of a different type. So the sense has changed, but over here nothing is formed, so no sense mutation. And that is what you see in beta thalassemia. So why has everything stopped forming? Because whatever mutation happened, that resulted in the formation of stop codons. What are the stop codons? UA, UAG and UGA. That is how you say it. UAA, UAG and UGA. Some change has happened that one of the stop codons has come, as a result of which no production will occur. So these are the 3-point mutation flashcard for all of you. Either it's not going to result in any change, so silence mutation, or it's going to change the entire sense, like in a case of sickle cell anemia, or it will stop the production altogether in a case of beta thalassemia. That is going to be because of a stop codon. So this was the first one that you had to do, point mutation. Now let's move on to the other one, which is known as a frame shift mutation. Now what is frame shift? Again, I'll write any random sequence I'm writing. You need not follow this only, okay? So now how did you read it? You started reading, what am I? You said ma'am is writing CAG, CGG, the CAG. You started reading alphabet, alphabet, alphabet. Body doesn't have so much of time. Body will read it triplet, another triplet, another triplet. So body will say that every frame, this is frame number 1, then this is frame number 2, then this is frame number 3, frame number 4. So body is going to read it as what? Every frame is gonna be a triplet for all of us, isn't it? Now I say that over here, one of these got deleted. G got deleted, guys. Now listen to this very carefully. I'm going to do the same thing once again in a while. If G got deleted, so you will say, ma'am, now the frame will shift. Means now the first frame has become CAC. Next is going to be GGC, now AGC, and so on. So can I say, if there is any deletion, of a nucleotide, the frame gets shifted. Similarly, if I would have added something. So for example, guys, if I would have added, I've got the same frame again for you. If I end up adding something over here like A, now what do you think is going to be your frame? The first frame is CAG, but the second becomes ACG, GCA, GCG. So has again the frames changed. So it could either be a deletion or it could be an addition of a nucleotide. Both of them are going to result in it, and that is a change in the frame, shift in the frame. So simple, frame shift mutation is because of a deletion or an addition of a nucleotide, and that is also seen in cases of thalassemia. So when you go back, guys, and study about your thalassemia chapter in pathology, you will see all these mutations over there. So we are done with the mutations, that is the point and the frame shift mutations. The same question comes in your microbiome also. Now let's go on to the real bacterial genetics. That is gene transfer. How does one bacteria transfer material to another bacteria? And there are 3 methods that you have to study. One is transduction, one is conjugation, and one is transformation. So I can see 3 very important diagrams over here which can explain this concept to you in a second. If you see 2 bacteria making a connection, a male and a female, so like this, there's a male and a female having a sex pili in between, that is conjugation. Conjugation will mean that there is some kind of connection that has formed. If there is a tadpole wala bacteriophage involved, if a bacteriophage is involved, it is transduction. And transformation is, bus, pick up anything, pick up any DNA. Direct entry of DNA, transformation. That's how you define it. Let's start one by one. I'm actually starting with this transformation because this is the easiest. You're just going to pick up any DNA. So let's start with the very first genetic transfer, and that is going to be transformation. Something is going to transform, and the first scientist who gave you this was going to be Griffith. So actually this was given as an experiment. I'll explain that experiment to you first. Griffith gave an experiment in Streptococcus pneumoniae. And you don't have to know the entire experiment. Listen to it for 2 minutes. Second year students might get a short note, but for NEET PG students, listen to this, that if this photo comes, you should know what they're talking about. No one's ever going to ask you the pathogenesis behind it. So first, what have they seen? You have seen, if you go back to your general microbacterial anatomy, by now you know Streptococcus P wala organism, Streptococcus pneumoniae, is definitely going to be a capsulated organism. And my concept tells me that whenever a capsule is there, this means there is going to be virulence. And that is what is going to be known as a smooth strain. So basically, when is the journey, bacteria ka kya kaam hai? Bacteria has to kill. So when is the journey for the bacteria going to be very smooth? You will say that, ma'am, when it has a capsule, it is very, very dangerous. Its journey is very, very smooth. So now what are the things that you're seeing over here? There are 2 things. Can I say that if I put a capsulated strain means capsule hai, to matlab virulent hai, matlab its life is very smooth, it's gonna kill. If I inject it, the mouse is going to die, right? On the other hand, if I put an unencapsulated, if I remove the capsule and that means the capsule is gone, matlab the virulence is gone. This means the bacteria's life is going to be very tough and rough now, and that means when virulence is not there, the bacteria will live. So we have ultimately realized that if the capsule is there, the bacteria will be able to kill the mouse. If the capsule is not there, the bacteria will not be able to kill the mouse. This was understood. Capsule hai, virulence hai, mouse will die. Capsule nahi hai, virulence nahi hai, mouse will not die. Now what do I do next is, I am taking the same smooth matlab capsule is there. I am taking the capsulated strain, but I have heat killed the capsule. So basically the capsule has been inactivated. I have applied heat and the capsule has been inactivated. You'll say, ma'am, once the capsule has been inactivated, this means the mouse is gonna live. The mouse shall live because the capsule has been inactivated, fine. But now I'm going to do a mixture. I'm going to now combine two things. I'm going to combine this strain. What is this strain? Number one, rough. What is rough? Rough means the capsule is not there. There is no capsule. So should the mouse die? No, the mouse should not die. I have mixed that with a heat killed smooth strain. What is this? Heat killed smooth strain means, smooth means that definitely the capsule is there, but its activity has been decreased. Its activity has been decreased because it has been heat killed. So you will say either ways, ma'am, rough ke paas to there was no capsule. Smooth wala had a capsule, but you had reduced the activity. So overall your mouse should have survived. Because individually when you used a rough strain over here, your mouse survived. Individually when you used a heat killed strain over here, your mouse survived. But when you're using them together, you have realized that your mouse has ended up dying. Why? Because I realized that both of them transferred some material to each other. Although this had a capsule and this did not, it ended up transferring the capsule to it. It ended up transferring the virulence factor to it. As a result, in the end, my mouse ended up dying. This is the experiment that was given by Griffith, and later on, Avery, MacLeod and McCarthy ended up rediscovering it. So now I can explain to you what is transformation. It is the transfer of free DNA. So that capsule which had just been heat killed, that capsule got transferred. So you will have this kind of a bacteria, for example, and that bacteria will say, I am any damaged DNA that I'm seeing in the environment, I'm just going to eat it up. So I am going to uptake any soluble DNA fragments. How? Through what route is it coming? It's coming through and through the cell wall. Remember that bacterial growth curve, lag phase, log phase and all, through which phase during it will it come? During the division wala phase. So if I ask you that this is the bacterial growth curve, when is it gonna come? It's gonna come in this phase, during the division wala phase. And for this, the bacteria should have a lot of competence. So it requires something known as competence factor. These are the only three questions you get. What is transformation? Transformation just means picking up something. Who explained it first? The Griffith scientist. In which organism? In Streptococcus pneumoniae this was explained. This is very important. And now tell me, through what will it come? It will come through the cell wall. In which phase of the bacterial growth curve? The log phase. What does the bacteria need? It needs competence factor. This is done. Let's move on to the next one. Transduction. What do I need for transduction? What is this tadpole shape thing that we have over here? So this tadpole shape is referred to as a bacteriophage. This tadpole shape is referred to as a bacteriophage. Not only tadpole, it can have many shapes. It can also be circular. It can also be filamentous. But what we most commonly study is going to be the tadpole shape. But what is it? I'm calling it a bacteriophage. Is it a bacteria? No. Bacteriophage is a DNA virus. So you are saying that, ma'am, for one bacteria to give material to another bacteria, you need a virus? Yes. If this hand is one bacteria, this hand is another bacteria. Bacteria, they want to transfer things between them. Whom are they going to use? Virus. So can I say bacteriophage will pick up something from this bacteria, deliver it to this bacteria? So bacteriophage is nothing but it is one of your food delivery apps. It is picking up the food from the restaurant, giving the food to the client, and that's about it. So that is exactly what we are going to do in transduction. Remember your Swiggy, Zomato, that is what is transduction. That is what is a bacteriophage. So now, remember this was first demonstrated in Salmonella. If you can learn it, good. But what you have to learn is the most common mode of drug resistance in Staphylococcus aureus is going to be transduction. I'll give you a list of all these most common modes. Only for Staph aureus it is transduction, not for the others. We'll do that final list in the end. Abhi ke liye yaad kar lo that Staphylococcus aureus ka, when you say na penicillin resistance and all, that happens because of transduction. So for that you need to know there are two cycles which I'm going to explain to you, right? Just get familiar with the names and then I'll go into the details. Can you see these tadpole like chota Swiggy Zomato people, right? These tadpole like people, bacteriophages. What have they done? Either they can see, just focus on the two words. There is something called a lytic cycle and something called a lysogenic cycle. When I say lytic cycle, that basically means that whatever bacteria they are going to infect, when they'll come out, they will lyse the bacteria and come out. When they have to exit the bacteria, they will break. That is lytic cycle. Lysogenic means that they are going to make a combination. They are going to combine with the bacteria. They are not going to break and come out. But just remember these two things. Lytic cycle means that they are going to break and come out, and lysogenic cycle means that they are going to combine with the bacteria. So all these ratta things I'm making you do so that I can tell you the details in a while. So remember, there is something known as a virulent bacteriophage. Virulent means very, very dangerous. So it is just going to break. It only follows the breakage wala cycles, lytic cycles. Temperate phage has two cycles. Temperate has two cycles. It can follow the lytic cycle also. It can follow the lysogenic cycles also. So if they ask you what is the T1 and the T2 phage of E. coli, T1 and T2 phage of E. coli are going to be very, very virulent. Whereas what is the lambda phage of E. coli? That is going to be temperate. Temperate means it can follow both the things. So remember these again, like I said, pele rat lo. So virulent is very, very virulent. It will only break the cells. It is going to cause lytic. Temperate phage will follow two pathways, that is lytic and lysogenic. Now let's go into the details. Ratta ho gaya. Now let's start with transduction. Transduction is of two types. It is generalized and specialized. Understand generalized. So what do I have over here? This is a donor bacteria. This hand of mine is a donor bacteria. It says I want to give something to a recipient bacteria. So in the end, whom you have waiting for you is a recipient bacteria. So if you have to give something from donor to recipient, whom are you going to call? Your Swiggy Zomato. So matlab, who have you called? You have called the bacteriophage. Let us see what the bacteriophage has done. First the bacteria. So what is bacteriophage? What was I telling you? Bacteriophage is a type of a DNA virus. Matlab bacteriophage has its own DNA also. So first I see that the bacteriophage has injected its blue color wala apna DNA it has injected. It has injected apna DNA. So the first step is injection of bacteriophage DNA. Injection of bacteriophage DNA into which cell? Into the donor cell. First it says, first you take my DNA, okay. Now you'll say, ma'am, you have thoda bacteriophage wala DNA, and of course the donor DNA is also sitting over here. So using all these DNAs, I can see new bacteriophages are formed. But during this, when there is new bacteriophage production, when there are new bacteriophage copies that are formed, we realize that there is something known as mispackaging. What is mispackaging? The bacteriophage, that Swiggy Zomato person had kept and come over here and put his parcel here. So he had to pick up this parcel only and go ideally, but he got confused. Kuch mispackaging ho gaya. Some of the Swiggy Zomato people took their own DNA. Some of them took the red wala donor DNA. So basically there's a mix and match of the parcels that has happened. Mispackaging has happened. So look at this. I have some bacteriophages which have apna wala blue DNA. Some bacteriophages have bacteria ka red DNA. There's a mispackaging happened. They got confused between parcels. So now can I say that because of this, it will end up going and giving this donor DNA to the recipient DNA, but that is what you wanted. Can I say the bacteriophage will now go and give the donor DNA, it is going to give the donor DNA to the recipient DNA. So it's very, very simple. If you learn it as the food delivery app, it's going to be so easy. It means that this is a food delivery app person, that is bacteriophage. He came and kept his parcel over here, food parcel over here. He thought I will take this in a while, but after some while he got confused. Full mispackaging occurred. All the food parcels got mixed up and he ended up going and giving the donor DNA to the recipient DNA. See, red and blue ka mixture ho gaya. The donor DNA and the recipient DNA have recombined and the bacterial transfer is done. This is known as generalized transduction. So tell me one thing. For these Swiggy, Zomato and people to come out, what cycle was followed? This is gonna be released by lysis. Basically, the bacteria has lysed. In order to release them, there was lysis. So they followed the lytic cycle. Now let me explain to you what is specialized or restricted transduction. Again, I will tell you the same story, that here guys we have a donor and here we have a recipient, and of course I am going to use the same food delivery app person, that is the bacteriophage. And the bacteriophage does the same thing. It comes and leaves its parcel over here. It came and it said, I am leaving my food parcel over here. But now you know what happens. Now it's not alag food parcels. Now all the food parcels get mixed into one box. Or you can say that there is a relationship that is going to happen between these parcels. Jo bacteriophage ka DNA aaya and the donor. In the previous one, they remained alag. All the parcels were alag. In this story, all the parcels are going to get mixed in one packet. All of them are now in one packet. You will say, Arey ma'am, all of them have fused. So can I say that there is a fusion of bacteriophage and donor DNA that has happened over here? And what is this fusion known as? This is now known as a prophage. This fusion of the delivery app wala DNA and the donor DNA, they have fused. This has resulted in prophage. But although all the parcels are mixed, I will have to separate them na. But can you separate food like this? Will you be able to separate all the food? Of course not. So some mixing will happen na. Some vegetarian food and non-vegetarian food will get mixed up. So now tell me, when there is a process of separation that will happen, that separation is referred to as induction. And what is induction? Please note, this can either be done by ultraviolet light, or it can be done by nitrogen mustard. So these are the ways by which separation will occur. But like I told you, look at this carefully. Has the separation occurred pura? Has the red and yellow separated as it is? No. Little bit of red and yellow mixture has happened. So can I say that this time the bacteriophage could not take its entire sample separately out? It had a little memories from the donor, and that is what I wanted. I wanted a little bit of the donor DNA to come into this Swiggy taxi so that I can get it delivered later on. And now you realize, look at these. Look at the new bacteriophages formed. The new bacteriophages that are formed are basically going to have the donor plus self DNA. And that is what you call as donor plus self DNA. Very, very, very important. Okay, now you will say that ये जो नया hybrid वाला Swiggy taxis बना है, they are going to go to the recipient and the donor DNA will be delivered. From these hybrid, the donor DNA is going to get delivered. So now when I have to simplify this entire story, listen to this one line. In generalized transduction, this step was सबका अलग था. The bacteriophage kept its DNA separate, the donor kept its DNA separate. So separate bacteriophages had separate DNA. But in specialized transduction, both these DNAs have ended up fusing. So the bacteriophages that are formed are going to have all fused DNAs, and that is what is going to go and get delivered to the recipient. So that is going to be specialized transduction. Now coming all the way back to the cycle that you had. So look at it. If a bacteriophage has to follow two cycles, you will say number one, the lytic cycle. Lytic means the bacteria will burst and all the Swiggy taxis will be released. Bacteriophage is going to be released by all the cell breaking, that is going to be lysis. And what is lysogenic? Lysogenic is where I am seeing a fusion. So you said that ma'am, you saw a fusion. You definitely saw a fusion over here, but after some time you said that they got separated, or जिसे आपने induction कहा था, and again they went into that same delivery mode. But what if there was no separation? What if the fusion got a vertical transfer? Imagine ये जो Swiggy taxi वाला DNA है and जो donor DNA है, if they basically got fused and that got transmitted to the offsprings, will the offsprings now start having new properties? Yes, it will now start having new properties, and that is known as lysogenic conversion. That is known as lysogenic conversion. So what is lysogenic in simple words? Lysogenic is fusion. This bacteriophage DNA and the donor DNA ends up getting fused and then ends up going in the fusion form to the future bacteria. That is lysogenic conversion. The bacteria are getting converted with new properties. So now if I have to differentiate between generalized and specialized transduction, can I say generalized generalized में only lysis हो रहा था. I was only focusing on the lytic cycle. And why was that happening? Remember, the packages were mispackaged. That is what I told you. Whereas specialized में there was lysogenic cycle happening, and it was because of that prophase and induction, the fusion and separation, right? There was going to be defective excision during the induction. Induction मतलब that separation phase में there was a problem happening. Now tell me, generalized transduction basically shows you the lytic cycles. Which phage shows you the lytic cycle? You will say all the phages can show me the lytic cycle. That is the virulent or the temperate. Whereas specialized, which shows you only lysogenic, is gonna only be by temperate phage. ये कैसे याद करेंगे? Because abhi, rather than learning it like this, I told you there are two types of phages. There is a virulent phage and there is a temperate phage. I told you virulent is very virulent, means it's gonna break everything. It is going to have the lytic cycle. The virulent phage will only and only have the lytic cycle, whereas I told you the temperate bacteriophage will have two cycles. It will have lytic cycles also, it will have lysogenic cycles also. मतलब 2 मतलब it will be seen in both the transductions. It will be seen जहाँ पे lysis है, जहाँ पे lysogenic है. So temperate is seen here also, temperate is seen here also. Better is you learn it like that. This is just a simpler way of putting it, right? So now coming back, so even if you don't learn, this part is not needed. You just need to know for generalized transduction, quickly tell me what is the phase that you had? Lytic phase. And what was the mechanism? Mispackaging. For specialized transduction, what was the phase you had? You had all that induction and prophage formation, and it was going to be a case of fusion. So matlab there was lysogenic happening. Now how do I put all this information into my bacteria? Which are the toxins which are going to get affected by this lysogenic conversion? Lysogenic matlab fusion. These are going to be A, B, C, D, E. So remember, A and C toxins of Streptococcus, A and C toxins of Streptococcus pyogenes. Then you have C and D toxins of botulinum, C and D toxins of botulinum. Then the second C is going to be for cholera toxin. The D is going to be for diphtheria toxin, and the E is going to be enterohemorrhagic E. coli. All of these are going to get transmitted by that fusion wala process. Repeating, the A for A and C toxin of Streptococcus pyogenes, B for the botulinum toxin ka C and D. C is going to be the cholera toxin, D is diphtheria toxin, E is going to be enterohemorrhagic E. coli. So transformation and transduction are done. We are going to move on to the last one, and that is conjugation. This was explained by Lederberg and Tatum, and this was explained in E. coli. And let me tell you, finally I can make that list. If I ask you that which is the most common mode of multi-drug resistance? The most common mode of multi-drug resistance in the bacteria is conjugation. So remember, the resistance ka transfer, resistance ka most common mode is going to be conjugation. However, now is the time for some examples and exceptions. So please remember, I have already told you, except Staphylococcus aureus. Staphylococcus aureus is going to be most commonly by transduction, the bacteriophage wala. And also I taught you in the beginning that Griffith experiment for Streptococcus pneumoniae, because in Streptococcus pneumoniae it was transformation that was the most common. It was transformation that was going to happen. For all others it's going to be conjugation. So the most common mode of resistance is going to be, resistance spread is going to be conjugation. However, for Staph aureus the resistance is spread by transduction, and for Streptococcus pneumoniae it is by transformation. So abhi ke liye let's come down to the most common method, and that is conjugation. Conjugation will occur when it involves something called plasmid. So pehle to understand the matlab of plasmid. This is what you know for a plasmid. Let me draw a particular bacteria over here. If this is a bacteria, guys, have I already told you every bacteria has what kind of a DNA? It has a circular double-stranded DNA, है ना? ये तो main DNA हो गया. This is the main DNA or the main chromosome that every bacteria has. I'm not talking about this. It also has something outside the DNA. What have I written? It has something extra chromosomal outside the chromosome, double-stranded circular part called plasmid. So this extra material, which is also a DNA, this extra material is referred to as a plasmid. So can can sometimes they end up fusing, ये भी DNA वो भी DNA, they say okay let us make DNA का combination. Sometimes that can happen and that is known as episome. But I'm not interested, I'm just telling you that as a definition. I'm not interested in that fusion. I'm interested in why am I studying this plasmid, what all can this plasmid do. So now let me tell you what are the types of the plasmid based on their transfer. So I will say this plasmid can actually go to different bacteria, from one bacteria to another. How? So for example, if this is a bacteria, this says I want to give my plasmid to this bacteria, तो जाओ, that is known as horizontal transfer. Plasmid from here wants to go to the other one, बिल्कुल जाओ, that is horizontal transfer. This bacteria says I want to give my plasmid to my daughter, to my future, that is known as vertical transfer. So please remember, we have two types of plasmids called conjugative and non-conjugative. Conjugative plasmids are the ones which are going to be transferred both vertically and horizontally. Horizontally basically means it's going to be bacteria to bacteria, and vertically means that it is going to be bacteria to offsprings, to the children. That is a conjugative. Conjugative मतलब everything is going to happen. Bacteria to bacteria भी होगा, bacteria to children भी होगा. But non-conjugative ones are only transferred vertically. Vertically means only from mother to child, only from bacteria to the offsprings. By offsprings I mean future generation. It is not going to get transferred horizontally. तो ये तो definition था. Conjugative सब कुछ show करेगा, non-conjugative is only going to be vertical. But what is this plasmid? Why am I obsessing over this plasmid? इसमें क्या है? There are different types of plasmid. What is this plasmid carrying? I'll give you an example. If this plasmid is carrying a fertility factor, maybe it is responsible for the gender determination, male, female, or maybe it is causing some resistance, it is causing the resistance to penicillin, okay, or it is causing some production of antibiotics, so some factor it is carrying along with it. Please note, plasmids can number one have virulence plasmids, means that like if this is the bacteria, this was uska DNA and this was the plasmid, okay, this is the plasmid. You say, ma'am, plasmid is carrying genes for toxins. If that gets transferred, bacteria will end up showing a lot of toxicity. Okay, virulence factors it can carry. It can carry fertility. They are known as fertility plasmids, means that will determine male, female. They can carry resistance, means they will make something resistant to penicillins or beta-lactams or any antibiotics. Or they can code for bacteriocin. Bacteriocin wala is known as the call plasmid. So remember, there is something known as virulence plasmid, something known as fertility plasmid, resistance plasmid and call plasmid, right? So now you will say plasmid to transfer ho jayega, but I will ask you what plasmid are you transferring? So my main concern is going to be about F plasmid. That is what we are going to study now. Listen to this. You can all see now, bacteria, so you have identified this is the main DNA that you have, and this is the plasmid DNA. And you know, right now I'm talking about F plasmid, means I am not interested in any toxins. I am only interested in the fertility factor. The fertility factor is being carried. So I am going to label all these plasmids with F. I am going to label all these plasmids with F. So can I say that because this bacteria has the fertility factor, it is a male? It is a male bacteria. And if it will not have the fertility factor, it's going to be a female bacteria. Get familiar with the terminology. If you see the presence of a fertility plasmid, male hai. Agar fertility factor nahi hai, female hai. So now I'm going to transfer. How can I convert a female into male? How can I convert F negative bacteria to F plus? And that is what we shall study. So everyone first label with me. Look at this. You will say, ma'am, this is the DNA. This must be the plasmid. And कौन सा plasmid? I will write it as fertility factor वाला plasmid. Fertility factor है. If fertility factor is there, this is a male. And on the other hand, I only see the DNA. I don't see any fertility factor. I can't see fertility factor. Fertility factor nahi hai. So this is a female. How do I make them conjugate? Can you see there is a line forming in between them? Can you see that? This is known as a conjugation tube, because you are talking about conjugation. Na? This is known as conjugation tube, which is also referred to as a bridge. This is known as a conjugation tube, also referred to as a bridge. And this is made of what? What was helping in attachment? I'll give you options. Flagella? No, flagella the locomotion wala tha. Pili? Yes, pili help in attachment. Can I say these are sex pili? These are sex pili. That is a male female के बीच में तो sex pili होगा है ना? So they are going to form, and what will happen through them? Through this conjugation tube or bridge, I will realize that this F plasmid says okay, I will copy paste करते हैं. I will copy and paste one copy in you also. See, no, it's not transfer. It's not that plasmid gets up from here and goes and sits there. No, it makes another copy and the copy is going to go over here. So both of them end up having one copy. Remember, it's not a cut and paste. It's a copy paste. This already had the plasmid. This will keep that plasmid. One copy, it will go and paste it over here. So can I say this started off as F plus, ये तो हमेशा ही F plus रहेगा. But in the end, I realized that this F negative female, she also got converted into an F plus. I copy pasted a copy. So now we are going to have some reactions that we'll be making all the while. When there is conjugation between F plus and F minus, both of them end up becoming F plus F plus. Simple. So in the end, both of them changed into fertility factors. This is the first scenario that we have. Now we are going to study some nice scenarios of breakups and patch-ups. So what is the second scenario that you have to know? Abhi, now you start telling me the terminology. Okay? So this is the DNA. Okay, every bacteria has a DNA and this is the fertility factor that we have. So tell me, F plus bacteria hai, F minus hai. Fertility factor is there. Ma'am, this is F plus and F plus is what? Male. Yahan tak samajh aaya? Theek hai. Before you jump on to female, you know what I've done? Remember, I was telling you that sometimes they want to fuse with each other only. Instead of going somewhere, they decide, let us make and get into a relationship. Yes, both of them decided we will get into a relationship. So this DNA and this plasmid, they decided कि चलो let us enter into a relationship. What do you call it? So when the DNA and the F plasmid get into a relationship, that is known as HFR cell. What is HFR cell? It is high frequency recombinant. Recombinant word is only telling me that something has recombined. That is known as high frequency recombinant, where the DNA and the plasmid decided to enter into a relationship. So now imagine this relationship person whom you are calling as the DNA and HFR wala cell is now going to make a conjugation tube with a female. Now tell me guys, who is a female? You will say a female is, ma'am, someone who doesn't have the F plasmid. Poor one doesn't have the F plasmid. So you will say, wait, I don't have the F plasmid here. I have HFR this time. So now what will happen? Now tell me something. Both of them are in a relationship. When someone is in a relationship, do they do solo traveling? No, na. They will say, now we will to travel together. I will also go and you will also come with me. DNA will also go, plasmid will also go. But kya hua? Little bit of the DNA went, little bit of the plasmid went, and if it's a relationship, they will end up having a fight. Midway, this conjugation tube broke. Relationship tha na, to midway while traveling they had a fight. So little bit of the DNA was going, little bit of the plasmid was going. Midway the conjugation tube breaks. Please remember, the conjugation tube breaks. So is there a complete transfer or is there an incomplete transfer? You've understood it already. Ma'am, trip is spoiled middle way. They've not completed their trip. They have not reached their destination. So there is an incomplete transfer that happens. That means, now listen to this very carefully. Did the female have her own DNA? Self DNA? Yes, the female had her self DNA. Now you will say that, ma'am, thoda sa donor DNA came, little bit donor DNA reached, and little bit of this plasmid reached. Little bit of everything reached. Little bit of this donor DNA reached, little bit of this plasmid reached, but everything reached little because after that they had a fight and the conjugation tube, this conjugation tube from here got cut off. Now will little convert it into F plus? Will that do it? No. To convert it into F plus, you need a full copy of a plasmid. So you will realize कि मैम ये तो बहुत messy situation हो गया. So many DNAs have come. क्या क्या DNA है? The bacteria has her own DNA. Then the bacteria has a little bit of donor DNA. Bacteria has a little bit of plasmid, but very little bit of it. So it's become a mess. You will call this as a recombinant female, but still F negative. इतना कुछ recombine हुआ, but she could not get the full fertility factor. So now tell me something. If I have to make the end result and if I ask you that if there is a conjugation happening between a high frequency recombinant and a poor F negative female, you will say, ma'am, high frequency recombinant to remained the same. It remained in the same relationship. I expected that this female becomes positive, but she also ended up being negative only. That too recombinant negative. That too recombinant negative because she did not get the full copy. So that's your take home message, that you wanted to make her F plus, but she did not become F plus. Now comes the last part of the story, but for that you will answer everything for me. What is this? You know, this is the bacteria ka apna DNA. This is the F plasmid. It has a F plasmid. So what is this? This is an F plus male. Okay, what do I see in the next? They have fused. Both of them have fused. Now what do you want to call this cell? They have entered into a relationship. Can I call this a high frequency recombinant? Ye bhi pata hai ma'am. But after every relationship comes a breakup. Next step, they decided to break up. The DNA said, I am getting separated. Plasmid said, I am also getting separated. But does a separation happen without memories? No, na. You take memories, you take your gifts also back. So this plasmid said, wait, I will take a little bit of this also. This plasmid also extracts a little bit of the DNA of the bacteria. So can I say this cell is now known as an F prime cell? This cell is now known as an F prime cell, जिसमें थोड़ा combination is there. Now when you make an F prime cell fuse with or conjugate with a female, who is a female? Female is someone who has her self DNA, definitely has her self DNA, but doesn't have any fertility factor. She's F negative. So now you are planning to combine F prime with F negative. So simple, copy paste hoga. This time this F prime will copy and will get pasted over here. This F prime will get copied and will get pasted over here. So can I say now my female has converted into F prime? So can I now put up a very simple table over here that if F prime will conjugate with any female, both of them will end up becoming F prime, F prime. So first you will have to do a summary with me and then we will go on to the next topic. I know it's a tough one, but now I'll give you a very simple hint. Pehle to tell me the terminologies. What is F plus? F plus is when the plasmid is there. What is F minus? When the plasmid is not there. What is HFR? HFR is when the DNA and the plasmid have combined with each other. That is known as HFR. What is F prime? When the plasmid has separated. When the plasmid has separated along with little DNA. Along with the DNA. Little bit of memories are also there. Terminologies pata hai? Now you will tell me reaction 1, reaction 2, reaction 3, and the easy way of learning it comes now. Whatever you will have in the beginning, if you have an F plus in the beginning and that is conjugating with a female, both of them end up becoming F plus F plus. तो जो भी starting में है ना, whatever the male is, unfortunately it's male domination over here, ठीक है? तो जो भी starting में है, that is gonna be the domination in the end. Similarly, if I say there was F prime male, you will say okay with the female, both of them will end up becoming F prime F prime. So this is a patriarchal society, ठीक है? यहाँ पे जो male है, अगर male F plus है तो end में सब कुछ F plus, अगर male F prime है end में सब कुछ F prime. But if the male is confused, relationship वाला मेल, high frequency recombinant, तब तो complicate होगा ना, relationships are complicated. HFR with F prime combining, that will keep the F negative as negative. If HFR with F negative, if HFR with the female is combining, then the female is not going to get any positivity, then relationship mein to it remains as it is. So simple, if it is F plus and if it is F prime, whatever is the starting, that remains the ending for both. But if it is HFR, then the female is not going to convert, and that is the entire summary of this pathway. Coming to, ma'am, are you only going to transfer the fertility factor? Can nothing else get transferred? नहीं, ऐसा नहीं है. I also told you that plasmids can carry something for drug resistance. So instead of this, instead of calling it an F plasmid, now I will call it R plasmid, because maybe it's carrying factors for penicillin resistance, methicillin resistance, or any other antibiotic resistance, vancomycin resistance. So this R plasmid, ये बड़ा सा R plasmid, you will say, ma'am, what have you made? You have made a very big loop, and that is known as RTF, resistance transfer factor. RTF, this bada sa loop that you have made is resistance transfer factor, and you know this is like a big ring. Many factors can sit under it, many R determinants, chota. This means one big ring can carry resistance for many antibiotics at the same time. This is resistance for penicillin, this is resistance for methicillin, this is resistance for vancomycin. So imagine there's a very big segment called resistance transfer factor. On that chota R determinants are sitting together. This makes it a R plasmid. Together this is known as our plasmid. So please note that this is a very, very dangerous situation. If a bacteria ends up giving an R plasmid, means at the same time it can give so many resistances altogether. So this is going to become very difficult to treat. What did I learn? Transferable drug resistance via this methodology is very, very difficult to treat, because you have no idea that in one go how many resistances have been transferred. Whereas in the beginning, remember I told you mutations, point mutation, all of that. So point mutation is something that you see in TB. I'll give you a very simple example. You must have heard there are some people who are rifampicin resistant in TB. Rifampicin resistance in TB is because of a single mutation in which gene? RPOB gene. Single mutation. तो मैम single drug का problem है. This is not going to be that dangerous. Single drug का problem है. Replace that drug. So can I say that mutation which occurs at a single point is much easier to treat, whereas this transferable drug resistance is transferring so many determinants at the same time that it is very, very difficult to treat in the long run, and that is a question that you have in the exam also. We are almost done with all these transfers and all of that. There are two miscellaneous topics that I wish to teach you in bacterial genetics, and the first one is known as restriction endonuclease. The second one is going to be CRISPR-Cas9. When I talk about restriction endonucleases, you can see the word, I say that endonuclease, it breaks something that is DNA. Okay, they are produced by bacteria for defense against foreign bacteria. Okay, what is this? So for example, if this is a bacteria, so only we are not worried with bacteria. Bacteria can get infected with another bacteria. There is another bacteria who's coming and saying, I will come and infect you. I will come and infect you. So what will this bacteria say? Bacteria will say, Arey, how can you come and infect me? I have something called restriction endonucleases. I will kill you. I will be able to kill you. So can I say that restriction endonucleases are produced by this bacteria over here? Why? For its defense against any other bacteria entering, right? Like you have antibodies now that if COVID virus will come, I will kill it. Similarly, bacteria has restriction enzyme so that if any foreign bacteria comes, it will kill it. Every restriction enzyme needs a cofactor, that is magnesium. There are 4 types of restriction enzymes that are there. Bacteria uses all 4, but out of these, which is the type that I am using for my drugs and all the genetic engineering going on, all the lab testings, remember type 2 is used for tests, that is genetic engineering. Okay, so now I don't know how many of you understood the concept of why you're calling it a restriction endonuclease. You said, ma'am, basically what is so special about this enzyme? Look at this bacteria, for example. This is any random sequence I have written. So you know what the restriction enzyme does? Restriction enzyme says, I will identify a restriction site. I won't kill the bacteria anywhere. I will identify a restriction site, and that restriction site is going to be very, very chota, just 4 to 8 nucleotides. And that site is a palindromic sequence. Guys, what is a palindrome? If you have Malayalam, if you read it from left to right or right to left, it sounds the same. Simpler words, mom, dad, if you read them from left to right or right to left, they are same. So those are palindromes. Palindromes are what you read from left to right, right to left, they are the same. So it may be AATT, AATT. So if you read them from left to right like this, they are the same. So this enzyme says, I will find that sequence in the bacteria which has palindromes, left to right to left, same, and that is where I'll go and hit. That is the mechanism of action. That is the function that it has. But please note one more thing. I don't know how many of you identified this by now, but you should have seen this restriction enzyme is killing any outside bacteria coming. Why is it not killing the own bacterial DNA? Why so? Did you wonder? So now I'll tell you the most important thing. How does the bacteria protect its own genome? I'll tell you something. See, for example, if this is a bacteria, for example, and ये अपना खुद का DNA है, this is the own DNA, and unfortunately some foreign bacteria has also entered into it, तो ये foreign DNA bacteria है. You will say, fine ma'am, I have a restriction enzyme. Will the restriction enzyme go and kill the foreign bacteria? Of course it will kill it, but will it go and kill the self DNA? No. It will go and say, Arey, this is my own DNA. I don't have to kill it. How did you get to know? Because there is a process known as methylation. the self DNA has done a methylation. That is how it's done its protection. So they ask you, this is not killed due to methylation. It says, I have done methylation, you can't kill me. Kill that bahar wala bacteria. Don't kill me because I have protected myself by a process known as methylation. So take home message points for a restriction enzyme. Please note, number one, these are produced by a bacteria. Why? Because they want to kill a foreign bacteria. Point number one, how many are they? There are 4 types. Which is the one that I use in genetic engineering? Type 2. What is the cofactor? Magnesium. How does it protect its own self? By methylation. And what is going to be the site for identification? The palindromic site is there. The next miscellaneous topic, or the last miscellaneous topic that I have to teach you, followed by questions, is CRISPR-Cas9. This story is, many students, I know you guys are finding CRISPR-Cas9 very as a dangerous type, but if you know the full form of CRISPR-Cas9, trust me, it's very simple. So I've written the full form for you, but I want to actually tell you in a better manner. CRISPR and then Cas9. So first I want to tell you that Cas9 is nothing but a scissors. Cas9 is nothing but a scissors. What is CRISPR? CRISPR को देखो क्या लिखा है? Clustered regularly. Something is going to be clustered regularly. So what I'll do, I'll draw a line over here and then I'll say there are some clusters. This is one cluster, this is another cluster, this is another cluster, this is another cluster. ठीक है, there is something clustered regularly. What are these clustered regularly? These are short palindromic repeats. Short palindromic repeats. Fine. So if you ask me what are these clusters, I've told you they are wo palindrome, left to right, right to left, same wala. These are the short palindromic repeats. I've understood. So simple. They are clustered regularly, short palindromic repeats. And what is I? I stands for interspersed. I is gonna stand for interspersed. So what is this? Interspersed. What are these? There are some spacers that are present. Clustered regularly, interspersed, or interspaced also they call it, short palindromic repeats. So now you will tell me, ma'am, ye spaces kya hai? You know, I have learned, don't write it down because it is not in the textbooks, only I have made that short form. These are screenshots. These are screenshots. So tell me something. If COVID entered into our body, did my body remember it? Memory rakha na that this is COVID virus. If it will come again, I have to protect. If bacteria comes, do you keep memory of a bacteria of an infection? Hai na? So similarly, what has happened over here is, this is a bacteria and this time it has been infected with a virus. This has been infected with a virus. That means the virus is coming, but the bacteria will say, hang on, I will kill this virus, I will take his screenshot and I will save it in its memory. So this is nothing but the viral elements ka screenshot that it has left, the memory. This is the memory of the viral elements that has come up in the spacers, so that next time if this bacteria or this virus will come and try to harm me, I will remember it because I have taken a screenshot of it, I have taken a photo of it and I have kept it in my memory. So if someone asks you what are these spaces are the memory of the viral, dead virus. So what are they going to do? They are first, can you see this? Everyone, I hope you can see this. There are chota spaces that they have created. These are screenshots. These are screenshots of different viruses. Green virus, orange virus, blue virus, pink virus. Different viruses were trying. Every time I was taking a photo and keeping it in my memory. Who was keeping it? That is the entire concept of CRISPR. But who is going to kill that virus? Who is the scissors who's finally gonna kill that virus? It screenshot to le liya, theek hai, but you have to kill that virus also, na? And who's gonna kill it? Cas9. That's why I said Cas9 is a scissors. So this is the entire concept of CRISPR-Cas9, which means that CRISPR-Cas9 has spaces in which whatever virus had come, I took a screenshot, I kept it in my memory, and how did I kill the virus? With the scissors. So can I say that CRISPR-Cas9 is bacterial defense? The bacteria is using it as a defense against maybe some virus infection, right? So when a virus was trying to infect it, or maybe some other bacteria was trying to infect it, so this is how it was doing its defense. So both these things that I've taught you right now, restriction enzymes and CRISPR-Cas9, both of them are going to be bacteria ka defense mechanism. I hope you guys have understood it. I'm not saying it's a super duper simple chapter, but I hope I've tried to simplify it as much as possible, which I'll get to know if you answer well. Okay, question 1. Transduction is what? What was transduction? That Swiggy Zomato picture. So it was bacteria to bacteria via some virus. What will I say? Bacteria to bacteria mediated by some virus, that is bacteriophage. Viral mediated bacterial recombination. I told you, it's going to be from bacteria to bacteria, but who is going to get it? That tadpole wala bacteriophage is going to get it. Let's move on to the next one. The cell in which the F factor carries along with it some chromosomal genes. मतलब पहले तो relationship हुआ, then breakup हुआ. Breakup होते time the F factor carried some chromosomal genes with it. What is that breakup known as? That is known as F prime. Next, the F factor integrates with the chromosome. So relationship है F factor and the chromosome integrate, recombinant. What is it known as? It is known as high frequency recombinant. Let's move on to the next. You have an 86-year-old male who's a known case of diabetes for 3 years, presented to the ER, complains of non-healing foot ulcer, complained that he has been taking different antibiotics for a long time and the medications had no results. Doctor suspected कि कुछ तो drug resistance हुआ है. Which of the following statement is false? You have to find the wrong statement. Let's start reading. In mutational drug resistance, one drug resistance at a time is seen. Yeah, 1 point mutation occurs. बिल्कुल ठीक. Let's look at the last point. High degree of resistance is seen with transferable drug resistance. Yes, transferable had that resistance transfer factor and that could carry many chota resistance are determinants. So I had lots of resistance happening. ये भी true है. Can a drug combination prevent mutational drug resistance? Yeah, because I have learned mutational drug resistance is because of one drug का problem. तो अगर combination use करोगे, it will be sorted. That is correct. Look at the next option. Virulence is decreased in transferable resistance? No, I have just studied transferable resistance is a very deadly resistance, means the bacteria is gonna become more virulent. This is the false statement that we have. Bacteria is going to become more and more virulent. Moving on to the next question. The restriction endonucleases is a defense mechanism in which the bacteria system against foreign DNAs is killed. Bacteria kills the foreign DNA. But how is it able to protect its own DNA? What was the process? It's able to protect its own DNA by methylation. Very good. Another question. Function of CRISPR, the last question that you have. Come on, what was CRISPR? CRISPR was, there was a bacteria and that bacteria was trying to protect itself against some viruses or other bacteria. So, bacterial genome to protect against viral infections? Yes. Is it bacterial genome editing to protect against human infections? No. We are not infecting the bacteria. Some virus is going and infecting the bacteria and that is why it was taking that virus ka screenshot, remember? So option A is going to be the correct answer. And on that note, we have wrapped up our entire discussion of the bacterial genetics also. What are we going to move on to next? So when you talk about general microbiology, you have 2 more chapters left, which includes culture media and sterilization disinfection. If you are in second year, you can study it right now. But if you're NEET PG aspirants, intern, post-intern, I would suggest you can now start off with bacteriology, because better is that you do culture media right at the end. It will give you better, absorption power. You'll understand a lot more things. Same with sterilization, you can take it up right at the end once you finished your entire syllabus. It's going to make things very easy. But if you're in your second prof, I understand you have a university exam also to give up, so you can read those chapters right now also. So that's what we have next, culture media and sterilization.